I'm Dmitriy Kepkiy from NCATS, National Institute of Health in US. I'm going to be talking about our tissue chips in space program. So let me start with what NCATS actually is. It's a National Center for Advancing Translational Sciences. It was created as one of the institutes at National Institutes of Health over a decade ago with their mission of developing and catalyzing innovative methods and technologies to advance development of therapeutics with the goal of basically bringing more treatments for more people more quickly. And why is it a problem? Obviously, there is over 10,000 identified diseases and only a few of them actually have known treatments and cures. It takes awfully long time to develop therapeutics, and there is a lot of failures in the clinical trials. And NCATS is carefully looking at all of those problems. And one of the problems that identifies that the clinical trials fail a lot due to the lack of efficacy and very often due to the toxic effects not detected in other models but observed in animals. And it's sort of dependent on the particular disease and organ, but there's a difference in the toxicities which is detected in animals with toxicities which are actually observed in humans, shown on the right here graph. So NCATS was very keen on establishing the tissue chips program over a decade ago, and it was focusing on 10 major organs and systems here listed. And I don't have to show to those audience. Obviously, there was single organ chips developed like the one from Emulate, and multi-organ chips developed. The example would be Hesper's 5-organ chip shown on the right here. NIH created the Tissue Chips Consortium with the NCATS as the driving force for this over a decade ago, and in 2012 through 2017 it focused on safety pharmacology, and there was a number of awards funded under this initiative. And then it followed with the disease models for efficacy studies program, and you can see it funded a number of awards in this category also. And one of this is Tissue in the Chips program, which is funded during that time with 9 awards total funded. And now it's actually funding the precision medicine centers clinical trials on a chip to inform clinical trials design and implementation in precision medicine. And along those lines, NCATS is very active in terms of building confidence in MPS systems. It's funded tissue chips testing centers and database center. And as a matter of fact, one of the 4s, which is currently on the street, and NCATS is going to be reviewing soon, the Translational Center for MPS with the purpose of qualifying MPS systems as drug development tools. So going to space, it has been known for a while that space actually, space flight induces some risks for humans. For example, the space radiation is one of the major risks associated with space flights. And it was observed that it induced the damage to the central nervous system, reduced motor function, and result in some behavioral changes. The microgravity certainly was considered a hazard for human, which resulted in some profound physiological changes. But also there is more like psychological effects on space flight, like isolation and confinement, induce some psychiatric changes in astronauts, especially in a long flight duration. So NASA was interested in this effects on the human body, and it found some studies on model systems, animals, and one of the actually very famous studies is twin study in space, where two twins were basically analyzed. One of them was on orbit for quite extensive period of time, and their fluids were basically analyzed for different biomarkers and different kinds of omics analysis. And the outcome of the studies was that there are actually potentially some profound changes in the human body when exposed to long duration flights. For obvious reasons, you can study as much a human body, and we need actually much more robust and informative models to more deeper analyze the outcome of the flights. As I said, there is some profound changes induced by microgravity, and they can be divided by early response for the small duration flights to medium duration flights, and the long duration flights less than over 6 months usually resulted in muscle atrophy, cardiovascular deconditional, GI disturbances, hematological changes, and decline in immunity, and renal stone formation. And once you look closely at those actually resemble the physiological changes observed during aging very often. And that was quite stunning observation, and Katz was quite interested in pursuing this. Going kind of more into detail, the age-related physiological changes, which and Katz was interested. They were observed in aging adults over 65 years, and the GI conditions were changing. There was a atrophy of secretion mechanism, decreased gut motility, loss of strength and tone of muscular tissue and supporting structures, cardiovascular changes, heart valve tend to increase, atherosclerosis, etc. There was respiratory changes usually associated with age-related disorders, renal decrease in glomerular filtration rate and plasma flow and also decline in immune response and visual activity. That's kind of conditions which are very often observed in aging population. And to study those and come up with therapeutics, it's very difficult for obvious reason, because some of those conditions take long time to develop, and it's very difficult to actually set up any clinical trials in order to find and develop therapeutics targeting those conditions. And therefore, Katz was actually quite interested in utilizing tissue chip program to try to model those conditions in space conditions under microgravity conditions. So that's how Chips in Space program at Katz came about, and it was basically a partnership with NASA and International Space Station National Laboratory through the Memorandum of Understanding between Katz and those organizations, and with the focus of coordinate effort in biomedical research using tissue chips. The goal was basically to develop tissue chips that model human diseases and conditions for pathology and of major organs and tissue of human body exposed to those extreme environments of space, and deployment of this on the International Space Station. And this partnership also included space and implementation partners, which provided actually unique hardware and services to kind of set up their MPS for the payload, launch them, provide support pre and post launch operation planning, different kinds of engineering integration and technical guidance. So those companies are listed here. So, as I said previously, there was a number of awards, 9 awards actually funded by NCATS, and shown here under the goal of modeling age-related diseases under microgravity condition and translate those to improve human health on Earth. And they cover different organs and diseases from immunosense to kidney stone formation, post-traumatic osteoarthritis, and cardiac dysfunctional sarcopenia, and gut inflammation. And I said previously, but that's, I think, very important point to make, that studying those human biology is otherwise would be very difficult for, here on Earth. So there was a number of flights which actually happened from 2018, and the most recent one was in March of 2023. And basically each project got to flight their tissue chips around 2 times to their space. And here's the photos of the the spaceship taken off at NASA Kennedy Center in Florida. And here's the astronauts working in on the International Space Centers with those tissue chips. So this program is basically just finished, and a lot of on-ground studies are actually undergoing currently, and their publication is starting to come out and being published in different journals. So here's the 2 examples of some of the publications coming out of it. But basically, during those flights, there was some very extensive metabolomic profiles changes observed, and very often the increase in oxidative stress and inflammation was observed for the tissue chips in space. There was some changes in the genes related to immune system, in particular to immune senescence. On the chemical side of things, there was DNA damage observed, mostly probably related to radiation effects. In general, for the cardiovascular chips, there was observed cardiovascular decondition and cardiomyopathy. There was change in the contraction rates detected for cardiovascular chips in cardiomyocytes in space. And RNA expression profile changes for muscle chips, which basically resulted in muscle waste and down regulation of muscle development of those muscle fibers in space. Transcription analysis showed that there was down regulation of transcripts related to myoblast proliferation and muscle differentiation. And those kind of results are initial results. There, as I said, there's going to be more publications coming out of it, but that's kind of first example of such a deep analysis on DNA level, what's actually happening with tissue chips and tissues in space. So another goal of this program was actually to develop the automation and miniaturization of tissue chips instrumentation for space flight, because very often the tissue chips occupies a lot of space and actually require a lot of operation power to operate them, and those are not conditions which actually would be suitable for space. So a lot of optimization in tissue chips instrumentation had to be done in order to automate them and miniaturize. And a lot of progress has been made in this regard. So as the outcome of these efforts, NCADS recognize their successes with those efforts, and currently there is a funding opportunity in the works for developing automated and miniaturized systems which would be open for small business companies. We are also participating in the longevity extension of 3D tissues and microphysiological systems for modeling of acute and chronic exposures of stresses. That's a collaboration and partnership between NASA, NIH, BARDA, and FDA. And the primary goal of this is to extend the viability and physiological function of tissue chips to a minimum of 6 months, with the goal of actually developing a system for the long duration flights, such as someone would envision flights to Mars, for example, or to Moon would take. Here's the projects which were funded under this initial program with NASA, and again, they cover a lot of different organs and diseases shown here, and all of them are showing very exciting projects in this regard of developing longevity extension of 3D tissues. So just to wrap up, there's a lot of opportunities for biomedical research in space. One is certainly to better understand human biology and do the disease modeling at the conditions which are actually not available on Earth. It provides opportunity to study radiation, health effects, and other space hazards. It's kind of facilitates development of advanced medical capabilities, in particular telemetry, robotics, and automation. And we're also quite interested in terms of developing capabilities for long duration flights, capabilities to do the long shelf life pharmaceuticals and real-time manufacturing. That's actually related to one other NCADS program. And overall, also in precision medicine, because that's what would be required for astronauts on orbit, and how they actually would be fit for the long duration flights. And sustainable food source, that's also of interest. And finally, biomanufacturing in space actually offers some unique capabilities in terms of creating homogeneous protein crystals, which could be used for many different applications. Stem cell and bioprinting of organs and tissues for regenerative medicine. And as a matter of fact, there was a supplement funded to Shrike Zheng group to create blood vessels to model premature vascular aging by using 3D bioprinting in space, which obviously offers some unique capabilities in the microgravity conditions. So you can actually create some bioprinting organs in the microgravity conditions, which otherwise you wouldn't be able to do on Earth. And just to show you that there's going to be another interesting meeting happening in Niagara Falls in Canada in August of this year. And there's a lot of interest in this meetings, and if you can, please attend. And I want to thank a lot of participating organization in tissue chips in space effort. As I said, it's a collaborative effort between NASA, FDA, BARDA, NIH Tissue Chips Consortium, and certainly Office of Special Initiative at NCATS. And I want to thank you for your attention.
Thank you very much for very exciting plans for the future. So there are a few minutes left, so very quick question. One or two, yeah, okay.
Thank you. I was wondering, have you looked at any expression profiles at common factors among all these tissues? Is there like a master factor, do you think? Could it relate to the circadian clock? Or something related? And the second question, you showed impaired differentiation, for example, of the muscle cells. Do you think this microgravity could be used to maintain quiescence of stem cells?
Yeah, the question was whether the effects of the microgravity we can detect on the RNA expression level to circadian rhythms, right? That's the first question. And we're not really looking closely at it yet. And you know, a lot of this data is still being analyzed on the ground. So you know, we'll certainly keep it in mind if something like that is actually detected. We'll keep a note of it. And another question about the stem cell proliferation. Yeah, there is reports that the microgravity conditions actually changing how the stem cells are differentiated and what's actually happening to them in space. And I think we actually need to do much more studies in this regard. So those studies so far have been quite limited. Thank you.